Tell us a little bit about your research and how DNA electrophoresis ties into it.
Okay, I work at Leeds looking into diagnostics and developing new blood tests. In the field these are called biosensors, and really biosensors are any device which can interact with a molecule in your body, so that could be a piece of DNA or a protein that's released when you have a certain disease. And if we can detect that, then doctors can see whether a person has an illness or not, or they might be able to see how far along that illness has got, such as a cancer or a degenerative disease. This typically works by putting a drop of blood onto a sensor and reading out a signal. So one of the most widely known examples of this is the glucose biosensor, which is used by millions of diabetics around the world, and it helps them check their blood sugar levels so that they can manage their own conditions a lot better and not have to go to the doctor so often. So that's kind of the context. As to where DNA electrophoresis fits in, I do work looking at how we can detect pieces of DNA. So in certain kinds of diseases, as I already said, cancers, small bits of DNA are released into your blood. Typically, at the minute, to see whether a person has these bits of DNA, you have to take their blood, you then have to isolate the DNA, amplify it up, and then using PCR, and then you have to cut the DNA using restriction enzymes before you run an agarose gel. And all of this is done to see if you can isolate the bits of DNA that are associated with the cancer or with the disease. This is a very expensive and long procedure, and I work in a lab where we're trying to get this to happen within half an hour. One of the big needs for this is that the NHS spends up to 30 billion a year on diagnostics. So if we can achieve this, it'll obviously be really good for lowering those costs, and then this can be money that can be spent on drugs and surgeries and actually making people better, rather than just trying to work out what's wrong with them.